we're going to get into body fluids. So what are these body fluids that we often have to deal with at crime scenes? We're talking about blood, semen, urine, and other physiological fluids. We're going to talk about blood and semen. Those are more common than others. Saliva would be another physiological fluid, but blood and semen is what we tend to come across most. So we're going to talk about that. And from that, you'll know how to collect other types of fluids as well. So we're going to cover these main topics within blood. We're going to talk about blood first, getting samples, preserving evidence from clothing, from a portable object, something we can pick up and take with us to the lab, how we deal with blood evidence that's on part of a non-portable object, how do we collect that? And then we'll also talk about stains on non-porous surfaces. A few things about blood stain first. Blood, as well as other physiological fluids, are fragile, and certain best practices must be maintained. So one of those things is we're not going to package blood-stained evidence in plastic bags. One thing you'd never want to put into plastic is blood-stained item or anything with dried blood on it even. Because once you put it into a plastic bag and seal that bag, it no longer has any air circulation. It doesn't have any breathing capability like in paper. And with blood stain, the material can decay, it can rot, it can mold, and DNA is degraded rapidly. So we want to make sure we put our blood stain evidence into paper bags, cardboard boxes, that type of a thing. We're going to talk now about clothing. If possible, collect the entire stained garment. So you have a shirt with blood stain on it, take the whole thing. Some agencies, they are really about not putting as stuff in the property room that takes up space. And so if you have something with blood stain on it, they may direct you to cut that blood stain out. After you photograph the garment, you cut it out and you submit the cutout. But hopefully your agency is able to at least take a shirt. For one thing, when you get into court, putting a shirt on a mannequin that has blood stain on it is very telling. Now I can see it if it's a mattress. If you have a blood stain on a mattress, they don't want you bringing in a mattress to put in the property room. But cutting out the blood stain would be acceptable there. So we want to collect the entire stained garment. Avoid altering the stain or transferring blood from one portion of the garment to another. That really takes into account when you have to put it in the package. Are you going to have to fold it? You want to fold it as little as possible, and definitely not fold it where it's across the stain, and not fold it so a stain could contaminate another part of the garment. So I will show you a slide in a while of a shirt and how to package it by putting butcher paper between the different portions of the shirt and between the folds. So we'll see how to do that. Be careful not to lose or contaminate any remaining trace evidence on the garment. So one of the things I will suggest is when we are collecting something with blood stain on it, like a garment, put down a piece of clean butcher paper first. Put your item on there, and then when you fold that all up and put it in a paper bag, any trace evidence that may fall off of the garment will be contained in that butcher paper. And when it gets to the crime lab and they open that up, not only do they have the garment, but they have anything that could have flaked off or fallen off, and not just blood-stained material, but hairs or fibers, whatever else might be there. Now, when we are collecting and transporting blood evidence, we need to avoid excessive heat. We also need to avoid moisture, water, and other liquids. And we need to avoid exposing the bloody evidence to strong light, especially UV light. So one thing you would never do is have a bloody item and say, well, I need this to dry before I can take it to the station. I'll set it out in the sun. No, because the UV radiation from the sun is going to degrade the DNA in the sample. So you're going to want to dry it, but not in direct light. Avoid touching, taking off gloves, or coughing, sneezing over or near evidence. As you are talking to other people, if you're near evidence, from your mouth, your DNA is spewing forth. When we are dealing with bloodstain evidence, we never say it's bloodstain unless we've tested it at the scene and we can say it's blood. There are other things that could look like blood and turn out not to be blood later when it's tested. So don't call something bloodstain unless you've already tested it and know that it is. So instead, what we want to do is call things a red stain, or you could say an apparent blood stain. If you're at a crime scene and you need to determine if something is blood, there are testing strips that will normally do that. When we need to document this evidence, we want to make sure that we document it as it was found. So we're going to photograph it. We're going to include a scale and identification label, one of the placard numbers for the evidence number. We're going to take one or more location photographs that show the object where it was found. We don't just take close-ups of evidence. We need to take a mid-range to show where in the scene it's located, and then our close-up. When we have this evidence, we need to label it. And so we're going to label a container to put our evidence in. We're talking about clothing now, so it's going to probably be a paper bag. Could be an envelope, perhaps. Could even be a cardboard box. But we're going to have our name, ID number, date and time, case number, evidence number, and the location where we collected it, and a description. And of course, every item of evidence gets its own unique number, and it's going to be the same number that we show in the photographs. It's going to be the same number on our property report, in our sketch, so there's no confusion. Under the evidence description, we're going to include the type of item. In our notes and then in our report, we're also going to include the location of the stain. Sometimes you're actually going to write that on the outside of the package to assist the criminalist who may be looking at your evidence. You're going to record whether the stain is wet or dry, the location of the item at the crime scene. What if we do have some damp blood stain or wet blood stain? Well, we need to dry it before we package it, if at all possible. So if the item's wet, you want to place it on clean paper and allow it to dry before packaging, or we could transport it to the station to put in a drying cabinet. The clean piece of paper should be something like butcher paper, and you lay it out where there's going to be nothing disturbed. It's not windy or anything like that where things can blow on your evidence. And then you lay out your evidence and just let it dry, let it air dry. The other possibility is to take it to the station to a drying cabinet. Notice on the screen there, that's a picture of a drying cabinet designed to dry evidence. What you do is put a clean piece of paper, butcher paper, on the floor inside the cabinet. So as things dry, if anything falls off, you have the paper and you just fold that in on itself and label it to go with that item of evidence. So as an example, if your item of evidence is number 6, that paper that you're going to fold up after it dries is going to be 6A. So you just give it a letter following the number, keeps them together, and it's not like a separate item of evidence that you're taking pictures of. It's attachment to that item. Now, these cabinets, the way they work is they've got filters in them, and then they just have air that circulates. Some of them you can adjust the temperature on, but again, you don't want to make them hot because that will degrade DNA. The better ones also have UV lights in them so that after you take your evidence out, you can turn that on for 20 minutes and it kills all the DNA that's inside the empty cabinet, so you can now put in new evidence. How do I get it from the crime scene to the lab if it's not thoroughly dry? Lay out some butcher paper, put your item on it, fold the butcher paper over onto the item and make sure it's thick enough so that blood doesn't transfer through, and then transfer that whole thing to the station. It may not even go into a paper bag, but get it to the station wrapped in that butcher paper, and then you can hang it up like that. Well, if you don't have a drying cabinet, just lay out that butcher paper and dry it on there. You want to make sure you keep the stain intact in its original form and not transfer it to something else or another part of the garment, and allow it to dry naturally without direct sunlight. After that object is dried, then you're going to collect and label the paper on which the object was dried. And again, if the item is item number 6, you're going to call that piece of paper 6A. What I might actually do is see if I can just go ahead and fold that garment over with more paper between the folds so there's no transfer, but keep it on the original paper that I dried the garment on, and fold that up and submit it. That way you've got it all together. Otherwise, if you're not going to do it that way, you're just going to put it into a bag. You need to save that paper that it was lying on and make it 6A. Now, what about storing these items? Well, ideally, bloodstained items should be stored in a temperature-controlled environment between 60 and 75 degrees with less than 60% humidity. And it's secure, dry storage area. And if there's a vent with the heater heating the room, you never put your sensitive evidence near any vents. Now, let's talk about the actual packaging, and this is where I get to show you a picture or two. Let's say we have an item of clothing, let's just say a shirt, and it has blood stain on it. Well, we need to document that. So we're going to take photographs, we're going to add it to our notes, we're going to put it in our property report, add it to our sketch. An item should be wrapped in clean paper when the location or pattern of stain or mark is significant. We want to protect that. We're going to fold the garment if we need to, and typically we do. Wrap the garment in clean paper. When folding that garment, we're not going to wad it up. We're only going to fold it enough to get it in the container. We're not going to crease where there's a stained area. And we don't want to fold it where it's going to disturb any stain that we're considering. So here we have a shirt. There appears to be blood stain on the shirt. So what we want to do is fold it so that we can put it into a paper bag. But as soon as you fold it over, that stained area is going to touch another part of the garment. So you put a piece of butcher paper between the garment layers as you're folding. And just keep doing that. It'll make your package bigger, but it'll keep all the surfaces from touching another surface. We're then going to put that item into the labeled container like a paper bag. And it should be large enough to allow the air to circulate around the object inside of it. You don't have a bag or a box that's big enough to put it in. You could wrap it in butcher paper, transport it to the station, and then find suitable packaging. Seal the entire opening with evidence tape. Write your initials and date across the evidence seal. Since this is a biohazard type of evidence, put a biohazard sticker on the package. That way, anyone handling our evidence will know that they should be cautious. That was clothing. Now we're moving on to portable objects. So let's say you have a blood stain on a plate, and now we can take the plate as evidence. When possible, take the item with the evidence on it. So we're going to document, when appropriate, wrap the object in clean paper, as long as you're not going to disturb the stain or mark. So that's going to help to protect that area. Sometimes what you can also do is wrap with butcher paper if an object is still wet and you cannot get a chance to dry it before you have to transport it. So we're going to position paper on a portable object to keep the stain or mark intact and to avoid transferring any stain. We're going to place that item into a labeled container such as a paper bag or cardboard box. Again, it should be large enough to allow air to circulate around the object inside of it. If the object's too big to fit in any packages, that's when we're going to wrap it in butcher paper, take it to the station, and then figure out what we can put it in. When we do have it in a container, we're going to seal the opening with evidence tape, write our initials and date across the evidence tape seal, and put on our biohazard sticker. Now let's say that we have a non-portable object. Maybe it's blood stain on carpeting. First of all, we document. As far as collecting this, it's important to collect the entire stained area if the shape of the stain is significant. So if the stain shows a pattern like a shoe print or a hand print, we want to collect as much of that stain that shows all of that pattern. So let's say that we have blood stain on a carpet and there is a shoe print in that blood stain. Cut out that entire area using a clean scalpel, a utility knife or scissors, and we're going to cut that large portion and also try to cut some of the non-stained area as well. And the reason for that is we need to have some non-stained surface that's the same as our stained area so that the lab can have a standard to go by. If there's so much blood that you're just cutting an area that's only blood stained, then you're gonna have to cut a second sample that's a little further away of the same carpet that isn't stained, and that will be your standard. Now, if the stain's been absorbed into multiple layers, like a carpet down to a carpet pad, you're going to cut out from each layer. Here we have an example photograph. On the top here, we see carpeting, and there's some circles drawn and item numbers next to them. Well, those happen to have blood stain. So what do? We then, in the bottom picture, see we have cut out a section of the carpet. And so we have item 8 and item 9. So we'll collect these as 8 and 9. And then we'll also cut the carpet pad underneath them. And then we'll call that 8A and 9A. Another thing we have to do is when we cut these samples out is save the orientation of them. So get out your compass, see which direction is north, and on the bottoms of the samples, draw an arrow to show which direction is north. That way, if we have to reposition these, either again at the crime scene or use it for reenactment purposes, we know which direction to and what position to replace these at. If the item is wet, we're going to put it on clean paper and allow it to dry before we package. And then we wrap it in butcher paper, put it in a paper bag or a cardboard box. I mentioned the control sample. That would be an area control sample or standard. That's where we are going to have part of that carpeting. As I said before, if your sample is large enough, you've already got it built in, so it's okay. Otherwise, you need to get a separate sample, and then you'd call that 6B and 7B if you called the carpet pad A. And how you do that, we just go ahead and cut it out like we did before, and we're going to also have orientation. Now let's talk about if we have blood on a non-porous object that we cannot remove as well. So let's say we have blood stain on glass, on a door. Well, what do we do then? Well, we're going to document it. We're going to prepare our packaging. And then if the stain is dry, we're going to moisten a cotton tip of a swab and use 2 drops of distilled water to do it. Make sure we don't saturate the swab. Two drops is plenty. And you never touch the tip of the water bottle to the swab. That's cross-contamination. And you just go ahead and then swab the surface. We're going to take that swab, and on the very tip of the swab, we're going to touch gently and firmly to the stain and rotate the swab. And then we dry that swab because we don't want to package it wet. If the stain is wet or it's damp, we don't need to moisten the swab. We can just go ahead and take the swab and get our sample directly. But then again, what we're going to do is touch the swab gently and firmly to the stain and rotate the swab and get that stain on the tip of the swab, not the sides. Then we'll dry the swab because that stain was wet in our drying rack. So what are these drying racks all about? On the right is just a little cardboard box that has holes in it where you can stand up your swabs. This is a commercially made one over here as well. It's actually a box with a plexiglass door, and it's got a plastic rack in there to put your swabs. And it does actually have a little fan. You could even take a styrofoam cup, turn it upside down, and stick your swabs in the styrofoam cup to let them dry. That's perfectly fine. Once that swab is dry, we're going to place the tip of the swab into a bindle. And we're going to fold the bindle so it seals around the swab tip. We're going to close the bindle and place it into an envelope large enough to allow air to circulate around the object inside of it. If the swab is perfectly dry, you could put it directly in the envelope. If no bindle or drying or swab drying box is available, use another sterile container that can hold the swab while it dries. So the control sample will be taken by using a second swab. You're going to put 2 drops of distilled water on it as well. And you're going to swab an unstained area of the same surface nearby. And you dry it in the same way, and you package it in the same way, and you give it an appended letter. So let's say that I do this swab of the countertop, and that's item number 10. I'll also do a control sample, and that'll be 10A. They go into separate bindles, and they go into separate envelopes as well. We're going to go ahead and demonstrate now. And the first thing I'm going to do is put on some gloves. And then also, because I'm working around this biological evidence, and I don't want to get any of my DNA on anything, I'm going to go ahead and put on my dust mask. So I've got that on. So what I want to do is get a sample here. Well, before I get the sample, I should go ahead and fill out my paperwork. I have a case number, the date, the time, and the item number. Description of evidence. Swab of red substance. Then the location where collected. On our property report, it's going to have the address, but I'm going to put bathroom tile. And then my initials or name and my badge number. The next thing I want to do before I actually collect is fold a bindle to put my collection in. So here is my glassine paper. And I'm going to fold this. So now we're going to have this all pre-folded. I'm ready to do my collection. All right, so now that I have filled everything out, I have everything ready to go. I have my swab nearby. I am now ready to collect evidence. So what I'm going to do is change my gloves. So this is a second pair of gloves here. All right, so now I can do my sample. So here I have the swabs, and these are sterile. So there you go. We're ready to make our sample. But I also need to have some distilled water. I'm going to go ahead and take one of the swabs out, and I'm going to put 2 drops of water on the tip. So now I have a moistened swab. So how do we collect? Well, basically, you just take the tip and you put it down on the surface and gently, firmly rub in a circle. And as I said, you want to make sure it's on the tip. And really, that's about all you need right there. It doesn't take a whole lot. And so now I have the sample. Now what I have to do now is let this dry because the swab is wet, and so is the sample because the swab was wet. So what I'm going to do is put this in a drying rack. So now I have that swab. Now what I need to do is a control sample. So I'm going to go ahead and take the second swab, put a couple of drops of distilled water on it. And I'm now going to get a sample next to the area that had the blood stain. And once again, I'm just rubbing the tip, rotating it to get whatever is there. And we don't see anything. So now I can put this one in there. So now I'm going to let them dry. Now, what you might be wondering is, hey, if I have 10 samples, how do I keep from getting confused about which sample is which? Because I'm going to have them all drying at the same time. What I have here are some Avery labels. And so let's say that this is item number 7 and the control sample 7A. So I just go ahead and mark them. And here is a sticker with a 7 on it. I put that on the stick. And then the 7A, put it on the stick. And now I can tell which one is which. So when they go into the envelope after they dry, or into the bindle first, then I keep everything straight. So now I have those drying. Let's go ahead and say that this is dry. But now what I need to do is get this inside here. How do I do that? Well, I'm just going to break off the tip. Now you could use scissors, or you can just break it off. But that's all you need is just the tip. The lab doesn't need more than that. So now what I want to do is put this inside the bindle. So I can open up the bindle, just drop that in there. Now make sure it goes all the way into the bottom. And then I can close the bindle. And it is nice and secure. Now I have the bindle, and you see it's not going to fall out. Now I have my envelope. I have my bindle with the swab in it. I'm going to go ahead and put the bindle inside. You don't have to label the bindle. You don't have to tape it. You just put it inside. And now you go ahead and secure that with your evidence tape. Come on, there you go. And notice that I have enough tape to wrap around the edges like that. And then the next thing I need to do is initial and date it across the paper envelope onto the tape. Because this is a suspected bloodstain sample, we need to put a biohazard sticker on the envelope. It's right here next to the word evidence. Liquid blood. So once again, before I make any collection, after I've labeled everything and I'm all ready to collect, I put on my gloves. And now I take my swab. And now the only difference is, because the sample is moist, I don't have to put on any distilled water. So I just go ahead and straight down on the tip, rotate. There is a very healthy sample. And go ahead and put it in the drying rack to let it dry. Then I would also do a control sample. Now for the control sample, what I'm going to have to do is actually moisten the tip. So I go ahead and I put 2 drops of distilled water on the tip. And now next to where that blood stain was collected, On the tip of this one, I just get a control sample. And then that goes in to the drying rack as well. When those have dried, then I go ahead and package them in a bindle and put them into separate envelopes. Separate bindles, separate envelopes. One more thing I want to show you. These are scalpels. Scraping and collecting. So I already have my bindle and I have a scalpel. So I'm going to open this. And so what I'm going to do is just scrape this dried blood stain and scrape what's in it into the bindle. So that's all there is to it. I mean, this is not rocket science, thank goodness. Now this will work on a smooth surface. When you are scraping on a textured surface, much more difficult. And that's where probably using a swab will be better. But you can scrape blood stain. It makes for a very good sample. And then just go ahead and scrape it into your bindle. Now, if it's on a vertical surface, you can actually kind of tape the bindle underneath. And then when you're scraping, it just kind of falls right into the bindle. You get as much as you can there. That's a pretty healthy sample there. But now in here, I have my sample, suspected blood. And you notice I did that with it all the way open. It's just a lot easier to do it that way. Now I can go ahead and fold this shut. And the nice thing, once again, is that stuff's not going to leave the package. It's in there, it's trapped, it's not going to fall out the corners because of the folds. Also detect blood in some circumstances. You've probably all heard of luminol. There are other products such as Blue Star, which is a brand name. There's fluorescein, which is a chemical, and they will do similar things. If you have blood at a crime scene that has been cleaned up, or maybe it's just traces and it's hard to see. Maybe it's not traces, but it's hard to see because the background that the blood stain is on makes it hard to see. You can go ahead and treat the area with luminol, and then the luminol or Blue Star or fluorescein will react with the iron in hemoglobin and it will fluoresce. So on the picture on the left is just a visible light, normal view. And then the picture on the right is what you get if you spray it with luminol and then turn off the lights. So this is a good way to detect blood. This is how you would find it. You could then sample, as well as you would photograph it, obviously, as you see in this picture, and then you would sample it.